Hello, everyone, and welcome to today's webinar, Testing Vaccines Final Dose Form to USP 787 Subvisible Particulate Matter. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Tony Harrison, Senior Marketing Manager, Beckman Coulter Life Sciences. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Tony, you may now begin your presentation.
Thank you very much for that introduction. Hello, everyone, and welcome to today's short webinar, about 25 minutes, on testing vaccine final dose form to the United States Pharmacopoeia Chapter 787 for subvisible particle matter. Now, before I start the presentation today, I wanted to share this quote with you. Of course, at the moment, we are all very aware of the current COVID pandemic struggle, and so I'm sure we can all resonate with this quote, and hence why I thought today's presentation was particularly timely. So as I mentioned, today's webinar will last approximately 25 minutes, and it'll provide a brief overview of why we test for vaccines for particulate contamination, compare and contrast the United States Chapters 787 and 788, explain why light obscuration is the preferred method for testing for particle contamination, touch on some of the challenges of using light obscuration counters for this application, and finally, explain how the HIAC 9703 Plus particle counter from Beckman Coulter is designed to overcome these challenges. So first, let's take a look at why we test for particle contamination. There's three primary reasons why we focus on particle contamination in parenteral products, such as vaccines. Firstly, these products are delivered by injection, and any particles present may block veins or capillaries, restricting blood flow, which could be particularly dangerous if they blocked the capillaries serving the brain. Also, particles could be carrying microbes, which would form potential threats in the form of infection. And lastly, it is possible that the foreign body itself could cause an allergic reaction, maybe even the highly dangerous anaphylactic shock in extreme cases. So now let's take a brief look at USP 787 and compare it to 788. Both of these are designed for particulate contamination testing in parenterals, but USP 787 is specifically targeted at therapeutic proteins such as vaccines. And of course, the manufacturer must specify which USP test they're going to release their product to, and this will of course be captured in the product marketing license. So let's take a look at USP 788 first. The primary method specified for carrying out this test in the pharmacopoeia is to use a light obscuration particle counter. And I'll explain the reason why this technology is preferred in a short while. Now, however, should the product under test not be suitable for light obscuration testing, then the USP does allow a secondary method of using an optical microscope and sizing and counting the particles by eye. Now, in order to achieve a reasonably statistically sound picture of the contamination levels in a batch of vaccines, the user is guided to pull at least 10 vials from the batch. That's if you're following USP 788. The user is then tasked with extracting and testing at least 4 times 5 milliliter samples from the pooled vials, i.e. a total of 20 milliliters, and plus the tare volume of the sample itself, itself. And for this reason, the total pooled volume from the vials must be greater than or equal to 25 milliliters. And I'll touch on the tare volume challenges in a short while. And for example, when vials of the final dose form may only be 1 milliliter, which is quite common, the user would have to combine not just 10, but a total of 25 vials to achieve the required 25 milliliter pooled volume. Now, results from the first of the 4 samples are discarded, and this is to try to reduce test-to-test carryover. And for small volume injectables with a dose form of less than 100 milliliters, the maximum particle counts per container is the pass-fail criteria, so it's per container. However, for larger volume parenterals of greater than 100 milliliter dose form, the pass-fail criteria is defined in terms of counts per milliliter, not counts per container. But in both cases, the particle sizes of interest are 10 and 25 microns. Now let's compare that to USP 787. Now the primary and secondary test methods defined in 787 are exactly the same. And the difference starts to come in where the number of vials are required. Due to the typically costly nature of vaccine products, USP 787 does not mandate the testing of 10 vials per batch. The only rider is that the pooled volume must be sufficient for the test. And likewise, the minimum sample volume allowed is just 0.2 of a milliliter instead of 5 milliliters. The user will still take 4 samples, still discard the first sample, but as long as the combined test volume and counter tare volume is as small as possible, the test could potentially be done just using 1 vial of 1 milliliter product. So that's different to 788. Like 788, however, the particle sizes of interest are still 10 and 25 microns, and the maximum per container are also the same. However, where the product is greater than 100 milliliters in dose form, the maximum allowable counts per milliliter and per container are both pass-fail criteria. If you remember, it was just particles per container with 788. So provided your product is a protein-based therapeutic and you can demonstrate compliance to USP 788, you can save costs and be less wasteful of the final product. Now, of course, chemically derived small molecule products must still be tested to USP 788. 787 is just for therapeutic proteins. So I mentioned several times there that light obscuration is the preferred test in both USP 788 and 787. And now I'll explain why that is. The problem was that particles contaminating our products are rarely conveniently spherical in nature. So how do you size them and compare them between batches when particles are so irregular in shape and you're counting them by eye, which was the previous method? There are several sizing conventions, but which is correct? And on top of that, sizing them by eye using a microscope is a very subjective process. And what we find is that technician differences in methods creep in, and as does human error. So some way had to be found to standardize the counting and to allow the methods chosen to be validated against certified traceable materials. Now, example of how human error can come in with a manual test here is shown by a study done by the Industrial Biodevelopment Laboratory, which showed more than 20% variability in test results between 16 experienced QC technicians all performing the same manual standard operating procedure. Now, clearly, the SOP might be able to improve and the technicians retrained, but such variability in results suggests that the manual process itself is not robust enough to deliver the required levels of quality control. And whilst the technical specifications of similar particle counting instruments may appear equivalent, when looking to try to improve quality control, QC teams should really be looking at the level of automation in the instrument, which will make the QC testing more robust and repeatable. Now, in a light obscuration counter, there's a light source, typically a laser, and which illuminates through a sampling window in the flow path and a detector on the opposite side of the window. And this detector turns the amount of light falling on it into an electrical signal, which can be measured. When particles pass through the measurement window, they partially block the light falling onto the sensor. And this reduces the amount of light falling onto the detector. The change in the detector electrical output is proportional to the size of the particle flowing through the laser light. Now, the attraction of this method is that the counter can be calibrated using certified traceable calibration beads of a known size. Now, these beads will obscure a fixed or certain amount of light, and now any particle, however irregular shaped it is, which obscures the same amount of light as the calibration beads, can be said to have an equivalent spherical diameter as the calibration beads. Thus, irregular shaped contaminating particles may be sized according to certified reference standards, and no matter what their shape, particles that obscure the same amount of light will be reported as being the same equivalent spherical diameter. Now, this helps almost eliminate the impact of subjectivity and human error from the measurement. Okay, so now you can understand why the USP prefers the use of light obscuration counters for these tests. However, there are some challenges of using this technology to fully achieve the benefits of testing to USP 787. So let's take a look at some of those. Now, as stated earlier, provide the counter is capable of precision counting in volumes as small as 0.2 of a milliliter, the whole test could be done on a vial containing enough for 4 samples plus the tare volume of the counter. Now, the tare volume is the volume of the sample pathway leading from the vial to the obscuration counting windows. In many general purpose counters, this tare volume is large enough to prevent the test being achieved with just 1 milliliter. The third challenge with light obscuration counters with small volume samples is the positioning of the sample probe inside the vial under test. Ideally, the sample probe should be vertical and central in the vial, especially if the vial is conical. The probe must go down as far into the vial as possible without touching the base of the vial, and the probe must not be allowed to get too close to the surface of the liquid in the vial, otherwise there's a very real risk of drawing air into the sample. And of course, air bubbles will be counted as if they were particles by the light obscuration counter, so you could get a false positive there, a higher count than you're expecting because the counter's drawing air. So really holding a probe by hand while sampling in a very small sample makes it very difficult to avoid these problems, especially if trying to complete the whole test with just 1 milliliter of product. The second challenge is whether the counter can achieve accuracy and precision or repeatability with sample volumes as small as 0.2 milliliters. So I'm going to use the target here to explain just what I mean. In the first picture, subsequent shots at the central bullseye result in hits all over the target. And this can be described as having neither accuracy nor precision. However, in the second precision, all the shots are bunched together tightly, but they're not hitting the central bullseye. Now, this is good precision, but poor accuracy. In the third picture here, the shots are all close to the bullseye, with 1 or 2 actually hitting it, but the shots are still scattered. And this can be described as having good accuracy, but poor precision or repeatability. And finally, in the last picture, this is what we want, where the shots have all landed in the bullseye, repeatedly demonstrating both good accuracy and precision. And this is the performance we need from our light obscuration particle counter. Okay, coming into the last section of our talk today. So let's take a look at how the HIAC 9703 Plus from Beckman Coulter helps users overcome these challenges. Well, firstly, the HIAC 9703 Plus gets around the tare volume challenges or problems by having a probe specifically designed to enable USP 787 testing, which has a tare volume of just 0.09 milliliters. Now, this should allow the user to perform USP 787 using just 1 milliliter of product. Secondly, the HIAC employs a motorized sampling arm controlled by a precision stepper motor, so it's automated. And this ensures that the probe is inserted accurately into the center of the vial and as close to the base of the vial without touching the bottom of the vial. Additionally, the HIAC has a vial clamp, which holds the vial in place during the test and ensures that the vial is centralized under the sampling probe. And this eliminates the variability that would be introduced should the technician have to hold the vial by hand. And thirdly, the precision syringe and stepper motor drive maintains repeat volume accuracy and precision on samples as small as 0.2 milliliter, which is the smallest sample volume USP 787 says we can use. And as can be seen in this table, testing on the sample accuracy of the HIAC 9703 Plus proves that the accuracy on samples as small as 0.2 millimeter is just plus or minus 1.02%. And whereas the precision calculation gives us a standard deviation of just 0.00205 on these samples. So the counting accuracy itself can be demonstrated using certified reference beads. And you can see here from these test results that the HIAC demonstrates greater than 90% counting accuracy on samples containing reference beads in just a 0.2 milliliter sample volume. So the HIAC is suitable for measuring samples using the smallest sample volume mentioned as permissible in USP 787, just 0.2 of a milliliter. So it requires 4 times 0.2 milliliter of sample for the test, plus the tare volume of the sample probe, which in this case is 0.8 mil of the sample plus the tare volume. Tare volume in the HIAC must be taken into account, as I said, which is 0.09 milliliter. And finally, you should aim to leave some of the sample in the vial once the test is completed to ensure that the probe does not pull air in the final sample. So say about 0.1 of a milliliter. Hence, with its automated sample holder, the vial holder, and high accuracy and precision with just 0.2 milliliter samples, the HIAC 9703 can perform USP 787 on vaccine samples with a total volume of just 1 milliliter, or slightly under, in fact. So that brings me to the end of today's short presentation on testing vaccines to USP 787, and thank you very much for watching, and we'll now open the session up for questions.
Thank you, Tony, for your informative presentation. We now have a poll question we'd like to share with you. It will pop up on your screen now. Your participation is greatly appreciated. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, please explain again what tare volume is.
Yes, certainly I will. So the tare volume is the volume of the sample pathway that the sample has to pass through up the sample probe, leading to the light obscuration counting window where the particles are counted. And in many general purpose counters, this tare volume is quite large. So it prevents the test being achieved with a small volume of sample.
Okay, thank you, Tony. Our next question is, please explain again what equivalent spherical diameter means.
Yes, certainly that's a difficult one, but it's very important really, and it's why obscuration is used. So the particle counter is calibrated using certified calibration beads, which are of course spherical, but they're of a known size, and they obscure a fixed amount of light in the measurement cell. Now, subsequently, any particle that enters the measurement cell, doesn't matter what shape they are, if they obscure the same amount of light as the calibration beads, they can be said to have an equivalent spherical diameter. And this means that we can count irregular shaped contaminating particles and size them according to certified reference standards, which gives us the traceability. And no matter what their shape, the particles that obscure the same amount of light will be reported as having the same equivalent spherical diameter. And of course, the main impact here is it helps to reduce the impact of subjectivity and human error on these measurements.
Okay, great, thank you. And our last question is, why do you say that holding a vial by hand when testing is not practical?
Yeah, so ideally, I mean, most particle counters, particularly with a single vial, tempting to hold it by hand, but ideally the sample probe on a very small volume, it should be vertical and central in the vial, and especially if the vial bottom or the base is conical, and the probe must go down as far into the vial as possible without touching the base of the vial, and the probe itself must not be allowed to get too close to the surface of the liquid during sampling. And remember, there's 4 samples to be drawn. Otherwise, there's a risk of drawing air in with the samples. If you get air in the sample, it's counted as particles, and that will skew your test results. So holding the probe, the vial by hand while the sample is tested makes it very difficult to avoid these problems, especially if you're trying to complete the whole test with just 1 milliliter of product.
Thank you, Tony. Do you have any final comments for our audience?
No, thank you very much indeed for your time, and thanks for watching, everybody. And I think we're all just praying for the arrival of the COVID vaccine, so that's why I thought this was topical today.
Yes. Thank you again, Tony, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.